Velkomin á Málstofu Hafrannsóknarstofnunar. Við erum heppin í dag því við fáum gest og það hann Björn. I'm not going to pronounce your second name. So. Schäffner. Thank you. Björn doktor í Sníkjudýrafræði og hann frekar víðförull með við flesta. Hann menntaði sig í Þýskalandi, fekk BS-gráðu þar. Svo fór hann til Tékklands, mastersgráða, svo Ástralíu, doktorsgráða og svo hann starfað í Brasilíu og Suður Afríku og endaði hér á Íslandi, þar sem hann starfar hjá Keldum. Vísindarannsóknir hans þverfaglegar og stundaðar í mikilli samvinnu við aðra aðila en í starfi sínu metur Björn heilsu vistkerfisins og áhrif af mannavöldum á lífríki sjávar með því að nota svokallað fornt hýsil sníkjudýrakerfi brjóskfiska og dýralíf þeirra. Hann sem sagt tengist Hafró á þann hátt að hann fékk að fara með í bæði haustral og vorral til þess að safna sínum og ef ég skila rétt þá hann að fara að tala að hluta til það hér í dag. So, Björn, the floor is yours.
Thank you. Well, I trust that Hlynur talked a lot of good things about me since I didn't understand it. But let's just hope that everything was correct. Yeah, welcome. Thanks for coming. I will talk about parasites as neglected ocean essentials. And usually when you hear the word parasite, it kind of causes this uneasy feeling, right? This, it's slightly disgusting. But why is that? Because parasites are simply organisms with a different mode of living. They need a host to survive. They live either in or outside the host. And their mode of living, parasitism, is in fact the most successful mode there is in nature. So every single animal in every single ecosystem on this planet is affected by parasites. And so take the giant panda, for instance. I know this is slightly off topic now from what you guys are doing here in Hafro. I will just take that as an example. So the giant panda has 35 species of parasites. So it is quite easy to assume that parasites might as well outnumber free living species. But it's not that easy to actually put the word parasite, the term parasite, on an organism. You are a parasite. No, you can't. You have to be very careful in doing so because there is the symbiotic continuum. So in nature, we have three symbiotic relationships, parasitism, commensalism, and mutualism, where either the host is harmed, the host is not affected, or both partners, the parasites and the host, actually benefit from their relationship. And as you can see here, I placed the mosquito under parasitism and the typical example of commensalism with the shark and their fish attached to it and benefiting from the food items that fall off. And then a tapeworm I put underneath mutualism where both benefit. And obviously, this might cause some uproar in the scientific world. How can you put a tapeworm under mutualism while tapeworm is the example of a parasite, right? But let's talk about more the sensu stricto term of parasites. And in nature, we have three major parasitic groups: the helminths, which are all the worm-like critters. We have the thorny-headed worms, the tapeworms, the flukes, and the roundworms. Then we have protozoans, which form the next clade of parasitic organisms, which are single-cell organisms, and arthropods, which includes the ticks, the lice, the fleas. Underneath you can see certain numbers. Those are all the different species that can affect humans. So you can see 305 helminths, 83 protozoans, some 50 arthropods, makes humans a very incredible host for parasites with 437 species that we know of. And of course, this also changes our perspective towards parasites. We consider them as something nasty, as malign disease agents that should at best be eradicated. However, if you consider all parasitic organisms, then a majority does not even have any zoonotic potential, so they don't do any harm to their hosts. And those are exactly the ones that I'm mostly interested in. And why is that? Why are some parasites more infective than others, and some more detrimental to their hosts than others? And that has to do with the time of coevolution. So us humans, as you can see here, we are on this planet for, let's say, 5 million years. That's simply not a long enough time for the parasites to adapt to us humans as hosts. The dinosaurs might have been a little bit better off. And then you have, for instance, cartilaginous fish with an evolution of 450 plus million years. So obviously the parasites that infect sharks and rays and skates and chimeras might not be that virulent as they might be for us. So on average, we can say that we see a decreased virulence in parasites the longer they have a co-evolution with their hosts. And having said that, in ecosystems, parasites even have certain benefits. So they are considered the key players for the ecosystem function, health, and diversity. And over here, I just list a few points. I will not go into detail there, but just to give you an overview about the positive aspects that parasites might have for their hosts and for the ecosystems. And one that I'm particularly keen on is the final two, which is most likely also the case for cartilaginous fish, that parasites are able to increase the survival rate of the hosts by helping them developing a functional immune system. Now that I talked a lot about the marine environment, let's dig right in. So in marine ecosystems, we can see that parasites are the major contributors, not only for the biodiversity, but also for the biomass, which most people might not even know about. So there has been a study done by Couris and co-authors in 2008, and this has been in an estuarine environment. So those authors found that looking at a set area of the estuary, they found that parasites are significantly higher in biomass than the apex predators, which in that case would be marine birds. So just implement that to the marine environment, which might be a similar picture but much harder to figure out, obviously. So parasites have an immense impact on marine ecosystems. However, they remain drastically understudied and neglected. And so this is where my research starts or grasps on. Especially here in Iceland, they seem to be vastly forgotten for the last 60 plus years. And so I try to implement a new project with the title Conservation of Ancient Relationships. Here you can already see that I put the word conservation in it, which also sparks some uproar in the scientific world because I'm talking about conservation of parasites, which might not be in the best interest for all the human doctors and scientists that work on human parasites. but I successfully implemented that already in South Africa and improved my base of collaborators that are listed here, all the different institutions that collaborate on that vast project. And coming here to Iceland, I also brought this project with and tried to implement it here. And Iceland has certain benefits. For one, we know exactly what kind of cartilaginous fish occur in Icelandic waters. What sharks raise skates and chimeras. Well, raise not a single one, but skates quite a few. And these organisms have been rarely assessed for parasites. So we have very limited knowledge about the parasite diversity here in Iceland. And as I mentioned just a minute ago, they are scientifically neglected. So in that Zoology of Iceland series, parasites have been listed, and that was written in the beginning of the last century to, let's say, mid-last century. And since then, not many people have actually focused on parasites, especially not from cartilaginous fish. So a lot of parasites that we might expect in Icelandic waters are not known, or the ones that are known might not actually be the ones that have been described from Iceland. So there is a lot to be done. Iceland itself has quite a few benefits, especially in the marine environment, because of the various oceanic inputs with varying temperature and salinity gradients, but also with the high species diversity. However, I think the biggest advantage for myself personally is the collaboration with you guys, because I was able to partake in 2 scientific trawls already and got a lot of nice material, which I will present in just a second. So focusing on the constricting parasites here in Iceland, that's like the major goal, right? In here, I want to assess all different sharks, skates, and chimera species that occur in Icelandic waters and observe the species diversity of their parasites. This will be then used, obviously, to verify those historical records where I trust there will be a lot of changes coming up in future. But also to check if there are any faunal differences in those different ecoregions in Iceland. For instance, the cold-watered north and east, or the more temperate south and west. So prior knowledge about parasites, as I said, is quite outdated, but so far the top contributors of parasites that we know of are cestodes or tapeworms with 17 species, followed by copepods with 11 species and monogenians, and an additional 3 species. So overall, we know 36 species of parasites that infect constrictions here in Iceland. Remember, the panda had 35 species, just to give you an overview, so the diversity seems to be very depauperate as of now. The top contributors for the constrictions are Amblyraya radiata, the starry skate, with a total of 12 parasite records attached to this name. Dipterus bartis, the common skate, with 9 records, and Somniosus microcephalus, the Greenland shark, with 5 records. So overall, 8 species of constrictions have been assessed so far, which only represents 19% of the Icelandic constriction fauna. As I mentioned, I was able to partake in 2 field surveys in the autumn survey in 22, where my colleague and I, Sam, which is on the bottom picture over there, we dissected a total of 97 animals. I will talk about the species in just a second. And in the spring survey, I was by myself, and I collected 95 animals of different species. So overall, I got 192 specimens belonging to quite a broad variety of species. And these ones I list over here. So overall, we got 8 species of sharks with 48 specimens, representing roughly 40% of the fauna native here in Iceland. Of the skates, we got 122 specimens, but have a look over here. Amblyraya radiata with 103 specimens, so this is the most dominant species in Icelandic waters. Other skates are certainly not that common, and we only got a single one of each. So I'm hoping in the next survey to improve on those numbers a little bit. So overall, 30% of the Icelandic skate fauna is covered. And the chimera species, we got 4, covering 67% of the fauna so far. But over here as well, you can see Hydrolagus only with a single specimen so far. So let us see what the future will bring. This is just to show you how those species look like. I will not talk about them in much detail, but each species that has been observed is illustrated over here. So for the collection of parasites, we focused on a broad variety of organs and organ types. I will only go for the preliminary results. I will talk about the spiral intestine, stomach, and the blood. All the rest that is listed here still needs evaluation, so I can't talk much about that. But over here you can already see a small variety of the specimens that I have detected. So the major groups, in fact, the major group is tapeworms, as one might expect, because the tapeworm diversity exceeds by far that of other metazoan parasite groups combined. So tapeworms diversified greatly in this host group, and as you can see here, they come in all different sizes and shapes. And at this point, I identified 23 species in 5 orders. Yet only 5 of those 23 have been reported from Iceland before. If the rest consists of new species to science that still needs to be seen. Then we also have other groups like the trematodes, monogenians, nematodes, and protozoans. Well, those species, they occur in much smaller numbers, as you can see here. Trematodes are flukes. This is a fluke over here. So it comes with 2 suckers, very easy to identify as a fluke. And they're fairly small, so you might not detect them in all the spiral intestine juice. And they are mostly dominant in teleosts, so sharks, rays, skates, and chimeras, they do not have that many species, which also explains why we haven't found that many. Nematodes, you probably have seen nematodes before in the field. We got thousands of nematodes. I didn't know what to do with them all, so I sent them all to my collaborator in France. Out of sight, out of your mind. Let's see with how many species she can come up with. So we have, at this point, no idea about how many species of nematodes we got from all those different skates and sharks and chimeras. And the monogenians is this group over here. They usually affect the gills or outer surfaces of constrictions. I haven't focused on the gills as of yet, so those monogenians I found in the rectum and in the body cavity. And there aren't that many species that occur there at those sites. So in that case, we found 3 species. I think that if we include the gills and observe gills of a broad variety of constrictions, we will end up with a much higher number there. And protozoans, blood parasites, we only found hemogregorines, which are these little bulbs here that are inside the blood cell. A single species was discovered infecting 2 different species of skates in Amblyraya radiata and Rayella phyllae. Now, both of them occur also in shallower waters. So there, the life cycle might be easier to facilitate because they are transmitted most likely by marine leeches. And the deeper you go, the less marine leeches you have and the less constrictions are affected by those blood parasites. This one here is a trypanosome. Until now, we haven't found a single one, but I continue looking because one trypanosome has been described from Amblyraya radiata. Now we are probably wondering what will happen with all of that material. Well, the idea is to collect as much material as possible and then just place it in the cupboard to make it look good. Of course not. So various different studies are in planning. Right now, we focus more on the biodiversity aspect. So we want to see how many species are out there, how do they differ in their morphology and on a molecular basis. Over here you can see tapeworms that infect sharks, rays, and skates. So you see this immense variety of different shapes and forms over there. And the same, as I showed you before, is visible here in Iceland, where they look different depending on the order that they belong to. So of course we want to assess how many species we actually have here in Iceland. Once we have this overview, it's also possible to track anthropogenic effects. Are there a lot of them present or not? What might be the reason if they're not present? We can assess the biogeographical dispersal of species in the marine environment. The evolutionary relationships, obviously, between themselves, but also towards their hosts, and cryptic diversity, which is coming up as a new issue, certain species that might be morphologically similar, but molecularly distinct. Then, of course, we will engage in some ecological studies as well, not only focusing on the parasites, but also on the hosts and on the ecosystem that this host-parasite system occurs in. And here we want to evaluate the host-parasite interrelationships. We observe changes in the population structures, potential faunal differences, and environmental changes in regards to evolutionary processes. The next study area will be ecotoxicology. This is something that I engage in quite recently. So basically using parasites as biological indicators for anthropogenic effects or faunal contamination in the ecosystem. So we can have a look at the host itself and then at the parasites, where we might also be able to assess whether these parasites actually have benefits to the host organisms or not. We know that certain cestodes and acanthocephalans are able to deposit a lot more environmental contamination in their bodies and thus might keep the host alive in highly contaminated areas. So we want to observe that. Especially here in Iceland, where we consider that our waters here are very pristine and clean. However, there are certain persistent organic compounds that might occur also in Iceland through ocean currents, and especially very long-lived species such as the Greenland shark is very interesting to observe. Now that I mentioned that, there have been studies ongoing with the Greenland shark, but here in Iceland this is quite limited, so this might be something to look into in future as well. Those pollutants might contain not only the organic compounds, but also heavy metals, in particular mercury, cadmium, and lead. So this is something that I will undertake with my colleagues from the Northwest University in South Africa and the University of Duisburg-Essen in Germany. And then this is an area that, as I mentioned before, is quite debated in scientific circles. I'm a very big supporter of including conservation science and linking that to parasitology, simply because there is an extinction crisis going on in terms of parasites that we don't even know of, since it has never been assessed. But if you consider 37% based on IUCN Red List, 37% of constrictions are facing extinction right now, and you can see how they declined since the 1970s. One might only assume what could be the case for their parasites, since every constriction has unique parasite assemblages attached to it. If this species is threatened, we are not talking about a single species, but a whole variety of species. And this has first been assessed by my colleague Chelsea Wood in the United States. And she went back to museum specimens dating back to the 1880s. And this is based on fish and fish parasites. So she assessed fish and saw how many species occur in the 1880s and then up until now. And she saw this drastic decline here in parasites that have a 3-host life cycle and a higher host life cycle, whereas the species with a 1 or 2 host life cycle remained pretty much the same. Well, bringing that to the cestodes, for instance, that occur in constrictions, 37% of constrictions would mean that about 2,000 cestodes are currently facing extinction out there. So this is quite shocking. I don't know how we are in the time right now. Second last slide. So what I am recommending is a paradigm shift. So we need to change our opinions about parasites and take them as the key players in ecosystems that they are. So we need to combat that parasite extinction crisis in order to protect global biodiversity. And so what we need to do there is firstly to assess all the negative impacts that we might experience if we lose the parasites in any environment. And then also consider parasites and any other symbiont assemblages as conservation targets in future. And with that, thank you so much for your time and yeah, for your collaboration. Thanks.
Okay, we have time for questions. If you have. I have, if you don't.
Please.
Thank you, Björn. Very nice. The biogeographical aspect of it. So are we expecting species to be differently distributed around Iceland? But that's the first question, and then there's a follow-up question, depending on your answer.
So in that case, yes, I would say so, because all those parasites that I introduced right now, especially the cestodes, they have a complex life cycle. So they are transmitted via the food chain, and you might have different organisms in different temperature zones around Iceland. So an Amblyria radiata that feeds on animals, invertebrates in the south and the west might have a different parasite composition as an Amblyria radiata that feeds on the north or the east with much colder influence from Arctic waters.
So saying that, you could then use parasites in migration studies of the fish?
Well, parasites are used in not only migration studies, but as biological tags for fish stocks. So, yes.
So they're like a stamp in your passport. You have been here.
So to speak,
yeah.
Yes.
Yeah.
Yes.
Interesting. Yep.
Yeah, thank you. Thank you very much for a good presentation. And it's, at least for me, it's new thinking about parasites.
Well,
the way you ended your...
I hope I could change some opinions here, yeah.
But I was thinking about, now we have this division at the University of Iceland, at Keldum, Department of Fish Pathology, or whatever it's called.
Yes.
Yes. Of course, they have been studying parasites in fish of commercial value, particularly in recent years or decades. But for sure, not the way you are doing regarding biodiversity, etc. But I'm just wondering, because you said that, well, you indicated that not much has happened here last 60 years or so regarding parasite research. But is it totally true? I mean, I think they have done some work at least. And even if it's, as I say, more focused on impacts on fish of commercial value and that kind of thing.
Yes. Well, I see where you're coming from. However, I talked about constricting parasites there that have been neglected for the last 60 plus years. And Árni Kristmundsson, for instance, from Keldum, he has focused quite substantially on fish of commercial value, which did not include constrictions. Yes. So yeah, there we have a much better overview, especially in the freshwater environment. We know exactly what occurs there, thanks to Árni. Constrictions, yes, a little bit neglected.
Any more questions? I have one more. So you're kind of Charles Darwin now. You're just listing species and just see what is there. But you talked about several projects that will follow on. Is it timed? Is it happening right now, or is it in the future, or
yes.
Will you need 10 or 15 years to do this? Or?
Well, obviously, those are long-term projects, yes. They are currently ongoing with my collaborators, and they're constantly changed and upgraded and with new ideas coming into it. But yeah, no, this is certainly ongoing right now. Yeah. And I wouldn't consider myself to be anything close to Charles Darwin, but no. Thanks for the compliment.
With that, I think we say just thank you very much, Björn.
No, you're most welcome. I also forgot to mention that I brought some parasites with. So if you're interested, have a look at those parasites that the ladies over there have. Thank you.